So I'm happy to introduce our next speaker. This is Delaney Fisher. Delaney Fisher is from the University of Virginia. She is pioneering the MRI guided focused ultrasound for the treatment of cerebral cavernous malformations, and she is here to provide some updates to her research.
Thank you so much for the introduction, and thank you all for being here today. I'm really excited to have this opportunity to talk to you all. So if you were here last year, you kind of got a glimpse into some of this research already, but I'm really excited to be able to give you some more updates that are pretty exciting. So I'm a graduate student at the University of Virginia, and we are working on using focused ultrasound, and specifically we're looking at a mechanism of focused ultrasound called blood-brain barrier opening as a new way of treating cerebral cavernous malformations. And today I'll be sharing some data that has been promising in showing that we can potentially control the growth of these CCMs as well as maybe even reduce the formation of new lesions. Okay. Let's see if this works. Okay. As I'm sure everyone in this audience is more than aware, the current standard of care for CCM treatment is surgical resection of the cavernoma. But of course, this is a very invasive process that can present risks to the patients and doesn't really address lesions that are not operable. And so our goal with focused ultrasound is to provide more non-invasive treatment options that are able to still target specific lesions and even treat those deep-seated lesions within the brain. And just to provide a little background of what focused ultrasound is, so this is a rapidly evolving, non-invasive therapeutic technology that has the potential to transform the treatment of many different medical disorders using ultrasound energy. And this allows us to target deep within the body, non-invasively, so without any scalpels or incisions, and to also do this without any radiation. And so most people probably have never heard of focused ultrasound before. I didn't either before I came to graduate school. And so you probably are familiar, though, with diagnostic ultrasound, which is a common method of being able to use ultrasound to produce images for different purposes, like monitoring pregnancies. With focused ultrasound, we are essentially just taking the transducer, which is what produces those sound waves, and curving it so that we can focus it into a specific small volume where we can then produce a wide range of mechanical or thermal effects for different therapies. And so I like to kind of compare this to taking a magnifying glass, holding it up to the sun, and concentrating those sun rays into a very small volume where you can then actually cause heating of like a leaf. So in the same way, we're just now concentrating those sound waves where we can then produce either thermal effects, but we can also produce mechanical effects. And specifically, the mechanical effect that I'll talk about today is blood-brain barrier opening. And to just provide some background here before we start talking about blood-brain barrier opening, I just want to make sure everyone is on the same page about what the blood-brain barrier is. So within the brain, we have lots of vessels, and these cells that make up the blood vessel wall are actually really specialized in the brain. And they prevent most things from actually crossing the blood-brain barrier, which is what we call it. So basically only very small molecules, certain drugs that you've probably heard of, can get across, and things like oxygen or sugar to provide your brain with different energy. So while the blood-brain barrier is often thought to be, something that you do want most of the time, it can also be a problem when we start thinking about different brain disorders. So you might have a really good drug, but it's actually not able to get across the blood-brain barrier to be able to treat that disease within the brain. And so with focused ultrasound, we essentially are trying to overcome this blood-brain barrier, and the way we do this is basically by using these sound waves that are within the focused ultrasound, and these sound waves are really just pressure waves. So within the sound wave, we're oscillating between a high pressure state and a low pressure state, and we take advantage of the effect that these pressure waves have on what we call microbubbles. So these microbubbles are just tiny little bubbles that are filled with gas, and these were actually originally used as ultrasound contrast agents to look at blood flow. And at the high pressure states of the ultrasound wave, these microbubbles will shrink because the gas inside is shrinking as well. And then at low pressure, the gas inside will expand within the microbubble, and so the microbubble will grow in size as well. So throughout this pressure wave, these microbubbles are oscillating between these high expanded states and smaller states. And specifically with blood-brain barrier opening, we're really interested in also paying attention to the amplitude of the sound wave. So the amplitude, and especially the maximum peak negative pressure, is really important as that can determine how much that gas inside of the bubble is going to grow, and therefore the maximum microbubble diameter. And the larger the bubble grows, the greater extent that we could expect with blood-brain barrier opening. And so to kind of see how this actually works within the body now, and kind of put all this into practice, here we're looking at one capillary within the brain, and we start by injecting these microbubbles intravenously into the bloodstream. And then we apply ultrasound specifically to one area of the brain that we're interested in treating. So of course, in this case, that would be the CCM. Then these microbubbles will begin to oscillate, so expand and contract wherever it encounters these ultrasound waves. And that causes this bubble to actually push and pull on the blood vessel wall, and we cause this temporary disruption of the blood-brain barrier. So now, if we were to inject a therapeutic agent into the bloodstream as well, it can now cross this blood vessel barrier and accumulate specifically where we targeted with focused ultrasound. And I also just really want to emphasize that this is a reversible and temporary effect. So we're opening up the blood-brain barrier to allow potentially a drug to get across, but this is only going to stay open for 4 to 6 hours, and we do expect that there's a complete closure by 24 hours after this treatment. And aside from just using this approach to look at drug delivery, it's also been shown that doing this blood-brain barrier opening approach can also help produce other beneficial effects to the brain. So one being the creation of new neurons, or neurogenesis. We've also seen that it can help to increase the clearance of different toxins that are building up in the brain and provide better transport out of the brain of these toxins. And it can also provide mechanical cues to the endothelial cells that can be especially important for CCM, where these endothelial cells are actually affected in this disease. And additionally, other groups have really looked at this focused ultrasound blood-brain barrier opening approach in Alzheimer's disease, and they've shown that this approach can be really beneficial. So in the case of Alzheimer's disease, other researchers have shown that you can have an increase in the removal of the amyloid beta plaques, which are a big component of Alzheimer's disease after focused ultrasound blood-brain barrier opening, as well as an increase in the neurons, a restoration of memory after this treatment, as well as activation of glial cells, which are the supporting cell types within the brain as well. So this has been done by other researchers, and I also want to provide a little clinical perspective here. So patients have undergone over 8,000 treatments with focused ultrasound in the brain as of 2022. And the large majority of these treatments have been centered on using the thermal effects of focused ultrasound to treat different movement disorders like essential tremor and Parkinson's disease, and also brain tumors. But there's actually been this rise now of using focused ultrasound and the blood-brain barrier opening approach that I'm talking about today in patients as well. And there's actually 25 different clinical trials right now that are ongoing or already completed looking at blood-brain barrier opening, with the large majority being for brain tumors, but there's also a smaller subset for Parkinson's disease and Alzheimer's disease. And so with all of this in mind, we really think that focused ultrasound could be a really promising approach for CCM treatment, and hopefully will provide new alternative methods other than just surgery for CCM. But before we can really do this, we need to make sure that this approach will actually work for CCM. So today I'll talk about some questions that we asked with our studies. So the first one is, will focused ultrasound, or I'll probably call it FUS on occasion, open the blood-brain barrier within the CCM microenvironment? We also want to make sure that blood-brain barrier opening is going to be safe, especially considering that these lesions are hemorrhage prone. And then finally, we want to know if focused ultrasound blood-brain barrier opening can actually produce this therapeutic effect in CCMs, kind of like what we see with Alzheimer's disease, so in the absence of any actual therapeutic drug delivery. And so to kind of walk you through our initial studies and how we answered this first question, I have here just a little schematic of how we approach our experimental design. So on the left here, we start by acquiring MRIs of our CCM mouse model, and this is specifically a CCM1 mouse model that we're looking at. And then we take pre-focus ultrasound images that allow us to choose which lesions we want to target. Then we will start to inject an MRI contrast agent, and then we'll perform our focus ultrasound approach. And here we're looking at a range of different peak negative pressures. And again, this is the amplitude of the ultrasound wave, which can affect how large those microbubbles grow during the sonication time, and therefore the extent of the blood-brain barrier opening that we might achieve. And then finally, we confirm blood-brain barrier opening by looking at the contrast enhancement after blood-brain barrier opening. So this MRI contrast agent that we're injecting generally does not cross the blood-brain barrier opening. So after focus ultrasound treatment, if we successfully open the barrier, we'll get this white signal on the tissue that allows us to confirm that we did indeed open the blood-brain barrier. There we go. Okay, so here we have some results from this first study. And I'll give you the spoiler now. We did actually see that we can effectively open the blood-brain barrier with our CCM mouse model, specifically within the CCM microenvironment. And to walk you through these results, we have here in the top row our high resolution or really precise MRI imaging that allows us to choose what lesions we actually want to target. And those are denoted here with these different colored lesions, and those colors match back up to the different pressures that we're using during treatment. Then in the middle row, we have our pre-focus ultrasound images where we have already injected that MRI contrast agent. So you can actually see a little bit of white signal on some of these lesions that are showing us just the baseline leakiness that these lesions have to that MRI contrast agent. And then finally, in the last row, we have the images that are taken immediately after focus ultrasound treatment. And here you can see there's an increase in this bright signal that's coming from our MRI contrast agent. And we actually see that as we increase the peak negative pressure, we also get an increase in this enhancement, indicating a better blood-brain barrier opening. And we're also seeing that this is telling us that we can increase delivery of our MRI contrast agent over the baseline leakiness of these lesions. So when you think about drug delivery, this indicates to us that we can actually increase the amount of drug that we might get to our cavernoma compared to just the baseline leakiness that a lesion might have. Okay, and then next we wanted to investigate, will this actually be a safe approach for CCM treatment? And so in order to do this, we can treat with focus ultrasound. We have a pre-image and a post-image 1 day later. And we use 2 different MRI sequences to evaluate this safety measurement. So the first one allows us to look at measuring the volume changes. This is a T2 weighted image. And then we also look at susceptibility weighted imaging, which allows us to look at the bleeding or the hemorrhage that could occur after this treatment. And we're looking at this before and after focus ultrasound treatment. And so what we see here is that on both the T2 weighted imaging that allows us to measure the volume changes, as well as susceptibility weighted imaging, which allows us to look at the bleeding or hemorrhage, we don't see any changes in our lesion volume before focus ultrasound, as well as 1 day after focus ultrasound for any of the peak negative pressures that we're testing here. And so that's quantified here in these graphs that look at the change in volume from the after to the before. So we see generally there's no increases and maybe even a few decreases with some of these pressures. And then finally, we wanted to know, will this actually be therapeutic? Can we get those results that we're seeing in Alzheimer's disease? So in these experiments, we actually broke up into multiple treatment arms. So we have some single focus ultrasound treatments where we're just treating once, and then we continue to image out to a month later. We also have a repeat focus ultrasound treatment where we apply 2 different focus ultrasound treatments and again, continue to image till a month later. We also, though, wanted to look at 2 different types of peak negative pressure regimens. So the first one is what we've already been talking about. So this is fixed peak negative pressure, where we're applying just the same pressure across for the entire duration of our sonication treatment. So again, that's the same as what I've shown you in all of the previous data so far. But we also wanted to look at this approach called passive cavitation detection, or PCD, modulated peak negative pressure. And so what this allows us to do is actually to take in account the microbubble activity during the sonications to adjust the peak negative pressure that we apply. So we might actually bump down the pressure as we go along if we notice any increases in our microbubble activity that might actually be dangerous. And so this is really important for us to include because this is the approach that will be used in the clinical trials that use blood-brain barrier opening. So we wanted to make sure that we also looked at this approach. And so in these different treatment groups, we first wanted to look at CCM growth. So we are measuring here the volume of the sonicated lesion that had blood-brain barrier opening with focused ultrasound. But we also looked at a lesion on the other side of the brain that did not receive any focused ultrasound treatment. And then we compared these two from before and after focused ultrasound treatment 1 month later. And so here we are looking at the single focused ultrasound treatment with the fixed peak negative pressure. And here on the top row, we have these contralateral lesions, or the lesions that were not sonicated. We're looking across time here from left to right. Then in the bottom 2 rows, we have the lesions that were sonicated with 2 different pressures that were applied here, again moving left to right through time. And what we find is that during this 1 month period after focused ultrasound, the lesions that we are not treating continue to grow in size, which is what we would expect in our mouse model. But the lesions that we actually target with focused ultrasound are actually controlled in their growth 1 month after focused ultrasound treatment. Looking now at the repeat focused ultrasound arm where we treat with 2 different focused ultrasound, we treat twice with focused ultrasound, and looking again at the fixed peak negative pressures with the same sonication throughout the entire, or the same pressure throughout the entire sonication, we again are finding that the lesions that we do not target continue to grow during this 1 month period, whereas we do not see any growth in our lesions that we are sonicated with focused ultrasound treatment. And here on the right, we have the different groups kind of broken down. So we have the non-treated ones as well as the different pressures that we're using. And we don't notice that there's any real differences between the different pressures that we're applying here. And then finally, we're looking at the repeat dose, but now with the passive cavitation detection modulated peak negative pressure, which is again what we would be using in clinic. So in this case, we again find that we are able to control the growth of our sonicated lesions, whereas the non-sonicated ones continue to grow during this time period. So overall, what we're seeing is that with both a repeat focused ultrasound or a single treatment, as well as our fixed peak negative pressure and this clinical approach, we're able to control the growth of lesions that we're able to sonicate with blood-brain barrier opening. And so next we wanted to see what effect could we have from blood-brain barrier opening on lesion formation? And so in order to do this, I first want to explain a little bit about the shape of the focal zone that we're applying when we do focused ultrasound. So if we look at the top-down view of the brain, you have a circle shape from the focal zone. But when we actually look between the eyes, this focal zone is actually more of like the shape of a grain of rice. So in a mouse brain, this grain of rice actually goes from the top of the brain down to the bottom of the brain. So we get this column of focused ultrasound opening. In a human brain, this would actually be much more precise. The grain of rice is much smaller, but in the mouse, it's actually a little bit large. But to be able to evaluate CCM formation, we first circle where we open the blood-brain barrier, so this column on the pre-FUS image as well as 1 month after focused ultrasound treatment. And then we mirror this column onto the contralateral side where we didn't have any focused ultrasound treatment. And then we're able to count the number of lesions that are existing before focused ultrasound as well as after 1 month after focused ultrasound. And that way we can compare, between the sonicated side, how many lesions are forming after 1 month, and then in the non-sonicated side, how many have formed in that condition. And so here we're looking at the results again from the study, and in the top row we have images that allow us to see where we open the blood-brain barrier. So you can kind of see that there's this white column in both of these brains in the top row, and that's where we're going to draw that circle for blood-brain barrier opening. And then we mirror that onto the other side where we didn't actually have any blood-brain barrier opening. Then in the bottom 2 rows, we have images that allow us to see the lesion formation either before focused ultrasound treatment or 1 month after. And what we find here is that we don't see any new lesion formation as a result of blood-brain barrier opening treatment. And we actually might see that in some cases we're able to even have this trend towards a reduced lesion formation when we treat with blood-brain barrier opening compared to the other side of the brain that did not receive focused ultrasound. Looking here at, so that was the single FUS treatment group, and now we're looking at repeat dosing. And we again see that we actually now have what's a statistically significant reduction in the lesion formation with our blood-brain barrier opening compared to the non-sonicated side of the brain. And now finally, looking at the repeat dose with the more clinical approach, the passive cavitation detection modulated peak negative pressure, we again find we're not inducing any new lesion growth as a formation of blood-brain barrier opening, but, sorry, I lost what I was about to say. And what's really important about this is that, we're actually seeing that this is an effective treatment. So we are not getting any new growth, and we might even be able to, with the right dosage of focused ultrasound and timing of focused ultrasound, be able to reduce the lesion formation, which could be really impactful. And so just to conclude here with this talk, this is the first time we've ever been able to apply focused ultrasound for CCM. So we are showing that, and again, this is in a mouse model. We also see that focused ultrasound is able to successfully open the blood-brain barrier, and it can actually enhance the delivery of our MRI contrast agent, which could be meaningful for drug delivery. We also see that this approach seems to be safe. It's not inducing any acute growth or bleeding after this treatment. And we also see that the blood-brain barrier opening can control the growth of sonicated cavernomas with one treatment, with multiple treatments, and the clinical or the non-clinical peak negative pressure approach. And then also that the repeat blood-brain barrier opening, specifically with fixed peak negative pressures, can reduce the formation of new lesions. And so just to give you an idea of what we want to look into next as a research group, we're trying to understand, why is this actually helpful? And so I have just a few ideas here, and we can talk about that more, but I kind of want to point out that we were asked earlier today about hemosiderin clearance, potentially, any drugs that could do that. Potentially, focused ultrasound could be helping us to increase iron clearance in that hemosiderin rim after blood-brain barrier opening, either through enhanced what's called lymphatic transport, or by activating some of these supporting cells that help to kind of eat up toxins that are building up in the brain. Also, we want to look at its ability to enhance the delivery of drugs, and especially drugs of differing sizes, so that allows us to look at more than just small molecule drug delivery, but also antibodies or gene therapies, and hopefully unlock whole new classes of treatment options for CCM. And then finally, I just want to acknowledge all the people that have supported this project. So we have my lab here on the top and my advisor, Richard Price, as well as our key collaborators on the project, so Peter Traverdic and Khadija Sharifi, who've created our mouse model that we're using, as well as Wilson Miller, who does a lot of our MRI support. And then finally, I also want to acknowledge our funding sources and also just highlight that the Focused Ultrasound Foundation is a really good resource if you want to find more. They have a website, and it's also very helpful. So I also encourage you to check it out there, too. And with that, I'm happy to take any questions.
How lasting are these results? Are these forever, or would you need repeat treatments over time, and what do you think might be that frequency?
Yeah, thank you. That's a good question. So right now, we're able to see, again, in a mouse, that this seems to be stable with one treatment for 1 month. The challenge now is to expand that to humans, of course, and we don't have an answer yet. So the good thing is that this can be a repeat treatment, that it can be done, and I hope that we're able to kind of look more closely at how many times do we actually need to treat, and can we get a better effect with a certain number of treatments, and that might help us to understand a little bit better how much repeatability this is needed and how sustainable of an effect we can achieve. Thank you, that's a good question. I think someone's about to bring you up.
So we've seen some of the results of stability and maybe possibly reduction. Were there any with some of the other trials with Alzheimer's and whatnot? Were there any side effects potentially with this, or?
Yeah, that's a good question. So I think I should look more closely at the Alzheimer's data, but I think there's only minor side effects of headaches potentially right after, some tingling, numbness in the arms immediately after, but all of those have seemed to subside pretty quickly after. So I think it's just more from stimulation of other neurons potentially as well.
Thank you.
Thank you.
What frequencies are you using?
Yeah, so in a mouse, we are using 1 megahertz frequency. That's pretty standard. But when we go to humans, we actually change that, and we use a 250 kilohertz frequency. The reason for that being the skull differences between the mouse and the human. So of course, a human has a very thick skull, and it can actually be pretty prone to heating. So we actually take a whole different approach when we look at the mouse versus the human application here. So with a human, you actually have this array, this kind of helmet that you wear that has multiple transducers scattered around. And then you spread out that ultrasound energy to just converge specifically where you want in the brain, and that helps to prevent a lot of the heating as well as the frequency changes. But in a mouse, we can use a simpler approach that's more affordable and more easy for us to use due to the skull being pretty thin.
And I saw MPA, is that millipascal?
Yeah, or megapascal.
Oh, megapascal,
Yeah.
okay, cool. Yeah, that's it, thank you.
Thank you. Thank you.